Before the 1920s, a diagnosis of type 1 diabetes was a terrible thing. It was a death sentence, especially for children. Doctors knew what was happening inside the body. They understood that the pancreas was not working correctly. This organ was supposed to make a special substance that helped the body use sugar for energy. Without this substance, sugar would build up in the blood to dangerous levels. The body would slowly waste away, no matter how much food a person ate. Patients grew thinner and weaker every single day, their bodies unable to find the strength they needed to survive. It was a slow and painful journey towards an inevitable end. There were very few treatments available, and none of them could cure the disease. The most common treatment was a starvation diet. Doctors would put patients on a strict regimen with almost no carbohydrates or sugars. This could sometimes extend a person's life by a year or two, but it was a miserable existence. Patients were always hungry, tired, and frail. They were living on the edge, with no real hope for a future. Families had to watch their loved ones, especially their children, fade away. They felt helpless as they saw the life drain from them, knowing there was nothing they could do to stop it. Children with diabetes often weighed very little, looking like skeletons covered in skin. Their lives were short and filled with suffering. They couldn't run and play like other kids. They couldn't enjoy a simple piece of cake on their birthday. Their world was one of constant measurement, restriction, and weakness. The hospitals had special wards just for these children. These rooms were quiet and sad places. The young patients would lie in their beds, waiting for the unavoidable end. It was a dark time for medicine, and a solution seemed impossible, a distant dream that no one dared to hope for. The medical community around the world was searching for an answer. Scientists and doctors knew the problem was in the pancreas, but they could not figure out how to get the helpful substance out of it. Every attempt to create an extract from the pancreas failed. The powerful digestive juices also produced by the pancreas would destroy the very substance they were trying to isolate. For years, this puzzle stumped the greatest minds in medicine. The world was waiting for a breakthrough, a miracle that could turn this deadly disease into a manageable condition and give hope back to millions of people. The story of this miracle begins with a young Canadian surgeon named Frederick Banting. He was not a famous scientist. In fact, he was struggling to start his own medical practice in London, Ontario. To make extra money, he took a job teaching at a local university. One night in October 1920, while preparing a lecture about the pancreas, he read an interesting article. The article described how a blockage in the pancreatic duct caused the main part of the pancreas to wither away, but left small clusters of cells, called the islets of Langerhans, intact. These islets were believed to produce the substance that controlled blood sugar. This article sparked a revolutionary idea in Banting's mind. He thought that if the main part of the pancreas, which produces digestive juices, could be made to wither away, then he could try to get an extract from just the islets. This extract would not be destroyed by the digestive juices. It was a simple but brilliant idea. Banting wrote it down in his notebook. Ligety pancreatic ducts of dog. Keep dogs alive till acini degenerate, leaving islets. Try to isolate the internal secretion of these to relieve glycosuria. He was excited and believed he was onto something huge. He knew he needed help and a laboratory to test his theory. Banting took his idea to Professor John J.R. MacLeod at the University of Toronto. MacLeod was a leading expert on diabetes. At first, he was not very impressed. Many others had tried and failed to find a cure. Banting's idea seemed too simple, and Banting himself was not an experienced researcher. But Banting's passion and determination were very strong. He kept pushing and asking for a chance. After some hesitation, MacLeod finally agreed to give him a small lab space, 10 dogs for his experiment, and the help of a student for the summer of 1921. The student assigned to help Banting was Charles Best. Best was a young medical student who was good at chemistry. He and another student flipped a coin to decide who would get to work with Banting first, and Best won. Together, Banting and Best formed a small but dedicated team. They had very little funding and basic equipment. Their laboratory was small and often uncomfortably hot during the summer months. But they shared a powerful goal to find a treatment for diabetes. They were ready to work hard and dedicate their entire summer to chasing Banting's big idea, an idea that could change medicine forever. The work began in May 1921. It was difficult and often discouraging. Banting and Best's experiment had several steps. First, they had to perform surgery on dogs to tie off their pancreatic ducts. This was the part of Banting's idea that was supposed to make the main part of the pancreas shrink. They had to keep these dogs alive and healthy for several weeks while this happened. This was challenging work, and some of the dogs did not survive the procedure. The summer heat in their small lab made the conditions even tougher, but they refused to give up on their mission. After several weeks, they performed a second surgery on the dogs whose ducts had been tied. They removed the shrunken pancreases, which they hoped now contained only the islets. The next step was to grind up these shrunken pancreases and create a liquid extract. This extract, they believed, would contain the pure, undestroyed substance they were looking for. They called this substance elatine. They worked day and night, carefully preparing their extract. Their entire hypothesis rested on the hope that this liquid held the key to controlling blood sugar and fighting the deadly effects of diabetes. Now it was time for the crucial test. They took another dog that had its pancreas completely removed, which made it severely diabetic. The dog was weak, thirsty, and its blood sugar was dangerously high. With nervous hands, they injected their precious islatin extract into the sick dog. They waited and watched, testing the dog's blood sugar levels regularly. To their amazement, it worked. The dog's blood sugar dropped significantly. The dog became stronger and more alert. They had successfully proven that their extract could fight the symptoms of diabetes. It was a moment of pure triumph and excitement. As their work continued, a lucky accident helped them move faster. They were running low on the dogs with the tied ducts, which took weeks to prepare. They needed a new source for their extract. Out of desperation, they decided to try using the pancreas from a fetal calf, which they got from a local slaughterhouse. They reasoned that a fetal pancreas was not fully developed and might not have strong digestive juices yet. This was a lucky guess. The extract from the fetal calf pancreas worked just as well. This was a huge breakthrough, a fortunate mistake that gave them a much easier and faster way to produce their life-saving liquid. With their success in dogs, the team, now joined by the skilled biochemist James Collip, worked to purify the extract for human use. By January 1922, they were ready for their first human trial. Their first patient was a 14-year-old boy named Leonard Thompson. Leonard was near death from diabetes at Toronto General Hospital. He was frail, weighed only 65 pounds, and was drifting in and out of a coma. His family had agreed to the experimental treatment as a last resort. The first injection of the extract, which they now called insulin, had a small effect, but also caused an allergic reaction because it was not pure enough. Collip worked tirelessly for the next 12 days to make the extract purer and safer. On January 23, 1922, they gave Leonard the second injection. This time, the result was dramatic and undeniable. Leonard's blood sugar dropped to near normal levels. He became more alert and stronger. For the first time in years, he could eat a normal meal without his body being poisoned by sugar. It was a true medical miracle. News of this success spread like wildfire. The hospital ward, once a place of quiet despair, was suddenly filled with hope. One by one, other children on the brink of death were given insulin and brought back to life. The discovery of insulin was a monumental event that changed the world. Banting, Best, Collip, and MacLeod became heroes. To ensure that everyone could get this life-saving treatment, they sold the patent for insulin to the University of Toronto for just $1. They did not want to become rich from their discovery. They wanted to save lives. Pharmaceutical companies soon began mass-producing insulin, making it available to patients all over the globe. A disease that was once a certain death sentence had been transformed into a manageable condition. People with diabetes could now live long, healthy, and productive lives. The impact of this accidental discovery continues to be felt today. Millions of people around the world rely on daily insulin injections to stay alive. The story of Banting and Best's summer project, born from a late-night idea and fueled by determination and a bit of luck, remains one of the greatest stories in medical history. It is a powerful reminder that a simple idea, when pursued with passion and courage, can lead to a breakthrough that saves millions. The discovery of insulin didn't just change the treatment of diabetes, it gave the gift of life back to countless families and offered a new dawn of hope in medicine.
Thanks for watching. Stay curious. If you enjoyed this video, don't forget to subscribe.